Hi guys. So this lesson is about environmental effects on phenotype. So amazingly enough, even though we've talked about all of these things and the gene says this and the gene says that, there are some genes that are actually influenced by the environment. So we call this concept phenotypic plasticity. For example, hydrangeas change color based on the acidity of the soil that they grow in. So the flower blooms are going to be different. So individuals with the same genotype will actually exhibit different phenotypes in different environments. So we have rabbits like the snowshoe hare and Siamese cats that at certain temperatures, they're going to have a lighter colored coat, and at other temperatures, they're going to have a darker colored coat. The soil pH for flowers like the hydrangeas that I was talking about. Also, we found that UV exposure can actually increase melanin production in the skin. It's called a tan. You get darker because there's more melanin being produced. Chromosomal inheritance and disorders. So not only can we have mutations on alleles, but we can also have mutations on entire chromosomes. You can have extra chromosomes, you can be missing chromosomes, you can be missing parts of chromosomes, you can have parts of chromosomes that are longer than they should be. All of these different things, these changes can cause, and typically do cause, a lot of disorder. So for example, Tay-Sachs is an autosomal recessive disease, and it has a mutated hexa gene. And when this mutated gene is present, the body will not produce an enzyme that breaks down this one particular lipid. It also will affect, we can have effects of the central nervous system, resulting in blindness because we're not breaking down this particular lipid. So this lipid kind of builds up in the system. Sickle cell anemia, autosomal recessive disease. We have a mutated HBB gene. There's literally one DNA letter that's different and causes all of this change. So it causes sickled cells that have abnormal hemoglobin molecules, and hemoglobin is critical. That's what carries our oxygen around. And then, like I said, we can have chromosomal changes. So for example, as we're going through meiosis and dividing up, what happens if the chromosomes don't separate correctly? We call this process non-disjunction, where during meiosis 1 or meiosis 2, the chromosomes don't actually separate correctly. This is one of the reasons we like to use karyotypes like what we learned about. So notice on the karyotype over here, what happened, we have an extra 21. So we call this trisomy 21, or of course, a lot more people are familiar with it as Down syndrome. There are 3 copies of chromosome 21. And remember how we talked about with the X chromosome that with females, because they get 2 copies, that a lot of times one of the X's, so the alleles will kind of go inactive so that you don't get twice as much. Well, think about in this situation, because it's not designed to do that, we have a whole extra chromosome, which means we have a whole lot extra of these alleles, which can then cause problems with the proper functioning of the body. Okay, so hopefully that has helped you with understanding that concept. Bye.